dear guests, dear colleagues, it is my privilege and great pleasure on behalf of EFSA to welcome you all to this breakout session on nutrition. I'm Valerio Courtouille, I'm the head of the nutrition unit at EFSA, and I'm delighted to have you with us to participate in this session, to share your knowledge, your questions, your thoughts, your ideas, and of course, to have some comments as well. I thank you very much for coming so early in the morning, and many thanks as well to all those who are following us through webcasting. We know very well that many of you have traveled very long distances to be here today, and that for us it's a big honor, but also obliges us and also reminds us about how important and relevant EFSA's work is. As we've learned, the motto of the conference is contextualizing risk assessment, and that means reflecting on the future of risk assessment in food safety while acknowledging the social and political context. In that spirit, the content of this session is constructed around two major questions. The first one is about where to focus the attention towards reducing the prevalence of diet-related chronic diseases. Is it single nutrients or foods and diet? We know that single nutrient deficiencies were the major public health concern in the nutrition area for most of the last century, and in consequence, the main target for nutrition science was on single nutrient diseases and related activities. However, at the end of the last century, the last decade, when the problems of nutrient deficiencies seem to be largely solved in Western populations, the prevalence of diet-related chronic metabolic diseases, such as obesity, cardiovascular diseases, and type 2 diabetes, increased steadily. And many scientists currently are calling for a shift in the nutrition research and for strategies, and in the strategies applied to reduce the disease risk. The second question is about how food innovation, societal and technological changes would impact modern diets and food choices, as there is a strong interdependence between food innovation, technological developments, and societal trends. The overall goal of the session is to explore how dietary guidelines should evolve to address the switch from nutrient deficiencies to diseases of excess as the predominant public health concern in developed countries. The organizing committee has put a lot of passion and effort in shaping what we believe is a very attractive program. We have with us top scientists who will bring their knowledge and experience in answering those questions. And we are very grateful to all of our speakers for the special effort that each of you made to be here today. The session will be co-chaired together with Professor Jane Woodside, who will also moderate the panel discussion at the end of the session. Let me briefly introduce Jane. She's a professor of human nutrition at Queen's University Belfast. She's a specialist in the use of biomarkers to assess dietary intake, in the conduct of human nutrition intervention studies, and a specialist also in interventions to promote long-term dietary change. A warm welcome to Jane, and thank you very, very much for accepting the role of co-chair and moderator. We will have two parts in this session. In the first part, we will have two debates on whether sugars and or saturated fats cause chronic metabolic diseases. And of course, these are not new topics in nutrition, are old-fashioned, we could say, but still are two topics that polarize a lot the public debate and public interest as well. For each of these debates, we invited two speakers who may have some different views, because we consider that listening to different arguments and opinions is a truly enrichment. So it's not going to be about what is right and what is wrong, but it's about getting more insight into the respective topics. After the coffee break, Jane will guide the discussion on strategies for dietary prevention of chronic metabolic diseases and the role and impact of food innovation and consumer trends. We warmly invite you to raise questions in the Q&A sessions and at the end in the panel discussion. You can raise your questions directly in the room or by submitting your questions through the application, as we have seen also yesterday in the plenary that it worked very well. So please feel encouraged to use also that possibility. And with this, I'm happy to introduce the first debate titled, Do Sugars Cause Chronic Metabolic Diseases? And for this debate, we have two well-known speakers, and we will hear today their expert opinion on the subject. I'm very honored to invite to come to stage Professor Graham McGregor, who will be the first speaker. Professor McGregor is Professor of Cardiovascular Medicine at the Wolfson Institute of Preventive Medicine and Honorary Consultant Physician at Barts and London. He's Chairman of Blood Pressure UK and recently served as President of the British Hypertension Society. He is founder and Chairman of the Consensus Action on Salt and Health, the World Action on Salt and Health, and Action on Sugar. He strongly advocates, and successfully we can add, for reducing salt and sugar in all processed foods and increase public awareness surrounding salt, sugar, and their effects on health. Thank you very much, Professor McGregor, and please come to the stage. The second speaker in the debate is from the other side of the Atlantic. I'm very happy to welcome Professor John Stephen Piper. Dr. Stephen Piper is Associate Professor in the Department of Nutritional Sciences and the Lifestyle Medicine Lead at the University of Toronto. He is also appointed as a staff physician in the Division of Endocrinology and Metabolism and scientist in the Li Ka-shing Knowledge Institute and St. Michael's Hospital in Toronto. His research is focused on using randomized controlled trials, systematic reviews, and meta-analysis to address questions of clinical and public health importance in relation to diet and chronic disease prevention. Thank you very much for coming, Professor Stephen Piper. Please come to the stage as well. Now, very briefly, the rules of the debate, which actually is sort of debate, is not exactly what we know to be a truly debate. Each speaker will have 15 minutes to present their arguments on the subject. We'll have question and answer session only after both presentations. We've heard both presentations, so not in between. And of course, in the discussion, the speakers will be invited to as well challenge or have questions to the other speaker. So with this, I'm very honored to give the floor first to Professor McGregor. Please.
Well, thank you very much. Chairs, ladies and gentlemen, a great pleasure to speak to you. I wanted to talk to you about sugar and NCDs. NCDs is non-communicable diseases, which is a terrible term, but it's by far the biggest cause of death in the world and includes cardiovascular disease, that's heart attacks, heart failure, strokes, and cancer, and various other things like accidents, road traffic accidents. And it's by far the biggest cause of death, not only in developed countries, but also developing countries as well. And when we look, and I'm sure you're all aware of the global burden of disease, that's a huge study that's going on, looking at what underlies the major causes of death, premature death and disability in the world. It's quite clear, and you'll see it here, that unhealthy food, that is food that's high in salt, fat and sugar and lacks fruit and vegetables, is by far the biggest cause of death in the world, followed by high blood pressure, which of course is also a dietary thing as well, mainly salt, and then followed by cigarettes. And of course we've been quite successful in fighting cigarette tobacco companies. You'll then see air pollution, then obesity, type 2 diabetes, which of course are rapidly expanding, will overtake these unless we get action, alcohol, and then high cholesterol, which is rather forgotten about, but is the major cause of furring of our arteries, atheroma, and all of us are developing atheroma, and that is the major cause of heart attacks, which remains in Europe the commonest cause of death and disability in Europe. And you'll see, therefore, that what we put into our stomachs and what we put into our lungs are by far the biggest cause of death, premature death, and disability. And yet, what do we do about it? We have taken action about tobacco. We've not taken action about the food industry, slowly poisoning us and killing most of us, and we haven't done anything really about air pollution. And it's time for action. Now, why does it kill you? Well, when you look at the underlying mechanisms, it doesn't require a degree or PhD in nutrition to realize that salt puts up your blood pressure, which you remember is the second cause of death in the world, and it causes strokes, heart failure, and heart attacks. Saturated fat puts up cholesterol, and that causes atheroma. And then, of course, fat and sugar are responsible for the very high calorie-dense fast foods, processed foods, but also some local foods. If you had some Parma ham, you'll realize how much salt is in those. And they have calories with no feeling of satiation and cause obesity, type 2 diabetes, and now clearly recognized is the fact that obesity predisposes a major cause of several types of cancer. And of course, sugar has the unique property of causing dental caries, which again has major implications for costs for society and looking after people with dental caries, particularly children. Now, why are we getting fat? Well, it's pretty obvious that it's due to the fast food industry. Wherever they move in, and the soft drink industry, people start getting obese. We go to China and India, the fast food, global food industry is moving in. What happens? Obesity, type 2 diabetes takes off. And this is the reason, as shown here in 2 American children who are grossly overweight. They're having a Big Mac, large chips, and Coca-Cola. Now, the calorie equivalent of that in fruit and vegetables is 11 bananas or 18 oranges. Well, you eat 18 oranges and try and have lunch. Whereas we could go now, although we'd have some difficulty in this desert round us, to find a McDonald's hamburger. We could have it now, and then we could have an Italian lunch, and we'd still be able to eat it. And this is the problem with this food. It only causes transient satiation. It doesn't fill you up. It has no residue in it. And of course, those people like Coca-Cola who feel they can burn off all the harm they do worldwide, these children have to run half a marathon to burn off what they've just eaten. A somewhat unrealistic prospect. Now, what is the impact of sugar? Well, we decided to set up an action group on sugar because we wanted to get a plan for the UK for obesity like we did for salt. And we're very experienced in how politicians work and how the food industry works. So we thought, let's take sugar because it's very media friendly. We can get huge amounts of publicity about sugar, but actually what we're really targeting is not sugar on its own, but it's getting a plan for obesity. So that was our aim. But of course, sugar is the only cause of dental caries, so that allows us to have huge publicity about the fact that all these children having dental extractions, commonest reason for admission to hospital, etc., and get big publicity about that. But it's obviously a major source of hidden calories, particularly in sugar-sweetened drinks and in confectionery, cakes, biscuits, sugar-laden drinks, coffee drinks and all those. And it obviously helps and leads to obesity and to type 2 diabetes. There's no disagreement. Everyone agrees about that. Where it gets controversial is, of course, does it, independent of its calorie intake, does it lead to obesity in some way? Well, clearly, there are several studies suggesting it may increase appetite. You may eat more if you get just the right level of sugar, fat, and salt together. But actually, when you look at properly controlled randomized trials, there aren't really any clearly demonstrating that. It could have an effect on blood pressure and cholesterol, and some of the sugar nuts think that sugar causes everything. But again, the evidence it puts up cholesterol or puts up blood pressure is very weak. Could it have some other effects? Well, clearly, it could directly make type 2 diabetes more likely. But again, when you look at the evidence, they're mainly cohort studies, follow-up studies, where there's no causal relationship. It's only in the nurses study from the US, women who had more sugar-sweetened drinks were more likely to get type 2 diabetes, but that doesn't show that it causes it. So very difficult to prove, but nevertheless a suggestion there. And then, of course, you've got the idea that sugar, particularly fructose, may predispose to fatty liver. But again, when you look at the controlled trials of outcome, there aren't any, and the actual evidence, except in animals given massive amounts of fructose, is not, in my view, very strong. But that doesn't matter, because sugar is an important cause of obesity. Whatever the arguments about these things, these are irrelevant to the fact that we want a plan to stop people getting fat and getting type 2 diabetes. So it's irrelevant to us and should be irrelevant to you, those of you who want action rather than debate and discussion. And we want action. It's too late now to discuss things. We want action. And this is what happens, of course. These very calorie-dense foods are immensely profitable. They cost nothing to produce. They're very calorie-dense, and they can make a lot of money from them. So it's very attractive to the food industry. And, of course, they only cause transient fullness or satiation. And they're everywhere. They're stuffed in your face all the time in brilliant marketing. Any time, you can buy McDonald's hamburgers any time of the day or night. They're everywhere, railway trains, aeroplanes. It's the same stuff, this rubbishy food pushed in front of you. And it's not surprising, particularly poorer people who don't seem to have the same control that you and I may have, become obese. And it's not their fault. It's the fault of the food industry and the soft drink industry. It's nothing to do with people. We're naturally programmed to eat more food when it's pushed in front of us and made to feel that it's filling some terrible need that you don't actually have by all this brilliant marketing and advertising. So what can we do? Well, firstly, we can tax foods that are high in salt, sugar, and fat, and that is coming in, as you see, all over the world. And there are various ways of taxing, either at source or on the product itself, and we're particularly in favour, and I'll come back to it, on a tax that involves reformulation, so that you can avoid the tax by making the food more healthy by removing the salt, sugar, or fat. We can ban unhealthy food advertising. Why do we ban cigarette smoking when in fact unhealthy food is a much bigger cause of death? And particularly to children, when you look at television, internet marketing, it's scurrilous the way the food industry markets their food to vulnerable children who have no concept of what a healthy or unhealthy food is, and then pester their parents to buy it. It is completely immoral what the food industry does to our children. And our policy is quite clear that all advertising, all marketing, all promotion of unhealthy food should be banned. Like tobacco, the food industry should only be allowed to promote healthy food and to market healthy food. We can restrict availability, which is pretty easy to say, difficult to do. We can reduce portion size, but by far the most important policy is to reformulate the food, slowly take out, because we can't ban these products, but we slowly take out the sugar, fat and salt, gradually making them more healthy over a period of time so people don't notice, just like we've done for salt in the UK. Most of the products that you buy in the UK have been reduced by 40% now in the last 10 years, with nobody noticing. The only problem is when you come to Parma and have a meal, it's completely inedible, which is a sad reflection on what's happened in Italy. Nothing. And of course, we also have to realise there's a big counteraction. The food industry are going to fight back, not surprisingly. And they also have a lot of advisors. And this is a paper in the BMJ about the web of influence of sugar. The people advising the government around nutrition, many of them are paid by the food industry. I'm not saying they're corrupt in any way, but you have to recognise these people are receiving money from the food industry and take that on board. And it's difficult sometimes to deal with, in the sense of how do you do with that when the government's being advised that sugar's not that important, there are other things, getting people to eat more healthily, telling them what to do and so on, which of course doesn't work. And this is a beautiful Times headline, because it had Coca-Cola paying millions of pounds to diet experts, and then probably the most corrupt individual in the world, Blatter, and Coca-Cola right in the middle. And you can make what you like of it, but I thought it was a superb headline to have Coca-Cola right in the middle. So we came up with a plan, having talked to our experts. What were we going to do? Well, we wanted to reduce sugar, so we said, right, we want a 50% reduction in sugar. You always ask for more than you're going to get, but we wanted a 50% reduction in all products that had sugar in, a 20% reduction in fat, focused on saturated fat, particularly palm oil, but other fats as well. And that would give us, we calculated roughly, 100 kilocalories per person per day reduction in the UK for each of those. We also wanted only healthy foods to be promoted and advertised, and a 20% sugar levy on sugar-sweetened drinks and confectionery, with the opportunity to reformulate. And that, we reckoned, very difficult to calculate, would reduce calorie intake by 50 to 100. So you've got, with that plan, you'd get a 250 to 300 kilocalorie reduction in dietary intake per day in the UK across, on average, the whole population, which would be enough to prevent obesity and type 2 diabetes. Clearly, we want public sector guidelines, schools, all that, and obviously we want uniform colour coding with signpost labelling. We're actually being sued by the EU because we have signpost labelling, unfair trade practice according to the EU, and we had a letter from the Trade Commissioner saying that trade was more important than public health. Maybe a reason why we're leaving the EU, not really. So the plan we got David Cameron involved, but unfortunately he resigned because of the EU referendum, and Theresa May came in, who's not nearly as forceful, but you can see we got a tax on sugar-sweetened drinks with the opportunity to reformulate. I'll come back to that. We got a 20% reduction in sugar of the 10 products that contribute most to sugar intake, and those targets have been set, but not a calorie reduction, only a sugar reduction. We're also setting targets for calories at the moment, which is code, of course, for fat reduction, and there will be probably about a 20% reduction in the calorie targets that are going to be set, although we're not quite clear how Public Health England are going to do that. And we have probably the strictest restrictions on marketing and promotion, but we haven't got a ban. But a ban will come in eventually. It will be quite normal in 10 years' time, just like it is for tobacco, not to see any adverts or promotion of unhealthy food. But it takes time. You have to be patient in public health to get these things. You can't get it straight away. The sugar levy is interesting because it should be copied by every other country in the world. What the Treasury, who are responsible for raising taxes, are much more intelligent than the Department of Health. They had a tax of 24p, 8% sugar. Most soft drinks are around 9%. If you got it below 8, you paid less tax. If it was below 5, you didn't pay the tax. And what happened is that apart from red Coca-Cola and the full sugar Pepsi-Cola, all of the soft drink, both the branded and supermarkets, have reduced their sugar levels below 5, more or less overnight, by using artificial sweeteners. We haven't got the exact figures because we don't have the sales, but we reckon it's removed hundreds of thousands of tons of sugar from the UK diet, which is an amazing achievement just in a few months. And everyone should be thinking about doing the same. So not just a tax to reduce consumption, but a tax to force the food industry to reformulate. And remember, the cigarette tax is 700% to 800% of the value. So eventually, a bottle of Coca-Cola in the UK will cost about 15 euros, which of course will have a huge effect on consumption. And Coca-Cola know it's coming and are preparing for it by diversifying into other drinks. So the message I have for you is it's time Europe took action. And maybe the European Food Safety Authority, the biggest cause of death is not infectious diseases or food poisoning, it's unhealthy food. Maybe they should take up that banner and start getting the EU to do something about unhealthy food, because at the moment the EU is totally dependent, subservient to the food industry. And it's amazing when you go to meetings how the food industry controls what the EU is going to do, and that's totally wrong. But the real message I have for you is that what we need to do is slowly make these unhealthy foods more healthy by reformulating them. And if it's done slowly, reduce the salt, sugar and fat and replace with complex vegetable residues, you can gradually and unobtrusively change the diet of the whole world and have huge effects on blood pressure, obesity and cholesterol with no need to change what people are eating and very little cost. Thank you very much.
Thank you very much, Professor McGregor. And we go straight to the next talk, Dr. Stephen Piper.
I get my first slide, please. Okay. Oops. So it's a great pleasure for me to come to Parma to share with you some of our thinking and some of our new data. We'll show some new data today in this area. I want to thank the organizers for the very kind invitation to debate Graham McGregor. I really enjoyed his presentation. I think you'll see some overlap and similarities that maybe we can talk about. So I've been asked to address the other side. So here are my disclosures. So put your filters on. I'm fully conflicted. So I've been trained by David Jenkins, so I put everything down from intellectual conflicts to financial and familial. My wife works for Unilever Canada. But I put in red those that I think that are most important. So what I wanted to do in walking you through the data is do it in a Socratic way, because it's a Socratic debate, and really do it by asking 3 questions and using those to inform the greater question about sugars and its contribution to chronic disease, cardiometabolic diseases. Are sugars independent of food form and energy linked with cardiometabolic outcomes? I think that's a reasonable question. What about sugar-sweetened beverages? Is this really the elephant in the analysis, as a lot of us think it might be? And what about other important food sources of sugars? Do they share and behave in the same way as sugar-sweetened beverages, and can we extrapolate from data from sugar-sweetened beverages? So let's take each one of these questions in turn. And what I want to do is invoke an evidence-based framework, a framework that's accepted, generally accepted universally by different systems of evidence-based medicine, that puts the greatest emphasis on those study designs which are used to inform public health policy and clinical practice guidelines, where really the starting point is our best designed observational studies from prospective cohort studies, and then randomized and non-randomized randomized control trials to give us the best protection against bias. And then considering that those are the units of analysis, then summarize those and synthesize those using the best tools we have to do that, which is systematic reviews and meta-analyses, which we see as quite separate, and so I've removed it here in this pyramid. And then apply an evidence-based framework for assessing that evidence using best practices with something such as GRADE, which we're starting to use. So if we walk through the data, starting with the prospective cohorts and trials, what do we learn about these questions? So if we start with the first question, are sugars independent of food form and energy linked with cardiometabolic outcomes? So what do we find? So let's start first with the prospective cohort studies. So we've conducted a series of systematic reviews and meta-analyses looking at this question in the prospective cohort studies from diabetes through cardiovascular disease. I don't have time to go through each one individually. We've published some of the data, and other data is just in process of being published, done by a lot of capable young students that I've pictured there. This work sponsored by the Canadian Institutes of Health Research principally, and Diabetes Canada and others. Sorry, I'm just waiting for this to forward. There we go. So what I've done is I've summarized the totality of the evidence that we have. Each one of these, this is a super plot of all the summary point estimates from those systematic reviews and meta-analyses going from diabetes to cardiovascular mortality. And what you can see, sorry, there's a bit of a delay here. Try again. What you can see is that generally we don't see an association. We look independent of food form and independent of energy in the prospective cohorts. We actually see a protective association for sucrose, which you can see there. Now, don't leave this talk thinking that somehow I'm thinking sucrose is protective for diabetes or cardiovascular disease, but I think it gives you some sense that there may be more to the story. We have to dig a bit deeper in terms of the food sources that drive that. Where we do see a signal, so the exceptions are for gout. We see this, and these are high relative risks, as you can see. There's some imprecision around it, but they're very high relative risk. And also for cardiovascular disease, for total sugars and fructose. But when we look a little more carefully, and we do a meta-regression modeling, so here it comes, what we see is that risk really starts at a high level of intake. For total sugars, 26% of energy, and for fructose, 12% of energy. Above where a lot of the population is, but where still a certain part of the population certainly is consuming, and so we have to be careful. But that risk that we see with total sugars and fructose as it relates to cardiovascular disease in the prospective cohorts appears to be restricted to very high intakes. Now, what about the randomized, non-randomized controlled trial evidence where we get a better protection against bias, but we don't have those public health important, patient important outcomes. So now we're looking at intermediate biomarkers. What do we see there? So we've done a series of systematic reviews and meta-analyses of randomized and non-randomized controlled trials from body weight through to fatty liver, sponsored by our Canadian Institutes of Health Research as the primary sponsor, and others that you can see there, done by a lot of really capable students. I don't have time to go through all the data, so what I'm going to do is to show you a summary plot. But first I want to say that the types of study designs that we found that inform this question around energy in particular, which is one of the points that I wanted to address with this first question, is we have two study designs. One, substitution trials. This is where the sugar, in this case fructose, which I'm going to show you, that thought to be that main bad actor in sugars, is matched in an equicaloric way with usually starch as the main comparator, so a isocaloric substitution. And that's, I think, a very relevant one because that's the one that we're going to see in the food industry through reformulation where the sugars are replaced with refined starches in the most part. And then addition trials where the fructose is added on top of, providing excess calories to a diet compared to the same diet without those excess calories. And you might argue that's where a lot of the population is, where they're over-consuming. But when we put those two together, how does that inform the question about the role of energy? So if we look first at the substitution trials, and again, I don't have time to go through all of the data individually, so I've summarized it here in a super plot, going from body weight through to fatty liver. When you match for calories, you do not see any adverse effect of fructose compared with starch. And I think, again, this is really important from the standpoint of reformulation. If you're just going to replace the sugars, in this case the fructose, with refined starch, we don't see any increase in body weight through fatty liver. We actually see a protective effect, if you like, on glycemic control and blood pressure, which is interesting because fructose was thought to be an important alternative sweetener even at the turn of the last century by Elliot Joslin, but we don't see an adverse effect. Now what about, and that's more than 50 trials and more than 1,000 participants, what about the addition trials? Well, then you see all the headlines. So where the fructose provides excess calories, then you see the increase in body weight, the increase in atherogenic lipid profile with increased ApoB, increased triglycerides, increased postprandial triglycerides, increased fasting glucose, increase in insulin resistance, increase in uric acid, and increase in fatty liver. But in the absence of seeing that effect under calorie-matched conditions, where the replacement is mainly with starch, this suggests that the calories are really driving the signal, which goes to what I think Professor McGregor showed in his slide, that the pathway through which the sugars are acting probably is a calorie signal. And it means that when it really answers the question, is there something special in terms of the metabolic and endocrine responses of sugars that leads to some downstream effects? The answer here really is that biochemistry, I think, is sound and the physiological effects are sound, but under real-world conditions, we don't see that it matters as much as the calories. The calories trump those effects. What about the second question? What about sugar-sweetened beverages? Well, if we look first, again, each one in turn, looking at the cohorts and trials. If we start with the cohorts, this base has been filled by a lot of systematic reviews, meta-analyses from groups other than our own. We've contributed one done by Jay, who's a second-year medical student now. He did this before he left for medical school. What you can see is when you look at this data, and again, I've summarized it here, this is an update of all of the most recent systematic reviews and meta-analyses where we've updated the data. Then you can actually see the headlines again. So we do see the increase in, this associated increase in diabetes, in weight, in hypertension, gout, and in CV mortality. So you do see these associations, and for a number of them, they're dose-response associations, so the quality of the evidence is a little bit higher, but although there is inconsistency in quite a few too. So the evidence is not strong. We can't infer causation, but there is a significant association. And this is really the evidence that drives a lot of our current guidelines and is invoked in a lot of those guidelines. Now, what about the randomized, non-randomized controlled trials? Well, actually, I can take you back to our systematic review and meta-analysis because the addition trials where the sugars were added to a controlled diet providing excess calorie compared to the same controlled diet really almost exclusively, with very few exceptions, derived from sugar-sweetened beverages. This is where fructose was put into the beverage and added on top of the diet. Then you see the increase in body weight. So this data really that I showed you is specific for sugar-sweetened beverages, if you like, if we're going to be honest about the evidence here. But the question is, does this data for sugar-sweetened beverages really hold for other important food sources? So sugar-sweetened beverages, in terms of a matrix, are just the sugar and water. There's no other nutrients there, no other fiber, no other components. Can we use this data to inform guidance as it relates to things like whole grain cereals that may have a bit of sugar, high fiber cereals, or yogurt, or fruit, because there are some people expressing concern about fruit and fruit products? So let's start first by looking at the prospective cohorts. So these are all new data. We've just, all of this is unpublished. We did publish an umbrella review as part of our systematic review on diabetes, looking at fructose-containing sugars independent of energy and food form. But all the data I'm going to show you is new data that our lab is generating, and we're just in the process of getting submitted and published. So if we look first at, so what I want to do is summarize them all. I don't have time to go through them all. If we look here at, this is for diabetes, looking at important food sources, we do see this association with sugar-sweetened beverages, with fruit drinks, mixed fruit drinks, not prefer 100% fruit juice. We do not see any adverse associations for any other foods. In fact, we actually see protective associations for things like fruit, whole grain breakfast cereal, yogurt, ice cream. Now again, don't leave here thinking that ice cream or a good Italian gelato is protective against diabetes, but this is the same prospective cohorts that have been used to implicate sugar-sweetened beverages. Here we see a protective association, and for chocolate. So I will be everybody's friend today. We see a lot of chocolate data coming out. But we don't see any adverse associations for these. It really is restricted to sugar-sweetened beverages. I'm sorry, the wrong presentation got uploaded because I wanted to go through some of the overall data. But what I'll do is, what I want to do here is just show you the cardio. So what I've done here, just to back up on my apologies, I don't have time to go through all the data, so I've just shown you the diabetes data. I'm going to show you the cardiovascular disease data, and it holds really across the different disease statuses. So this is now for incident cardiovascular disease, cardiovascular incidence. This is a very large systematic review meta-analysis involving more than 100 cohort comparisons on aggregate more than 5 million individuals with more than 181,000 cases of incident cardiovascular disease with an average follow-up of 14 years. What you can see here in looking through cardiovascular disease incidence, CHD incidence, and stroke incidence, we do not see an adverse association for anything other than sugar-sweetened beverages. So we do see this association for sugar-sweetened beverages that we've seen in the past. When we look at other important food sources, we do not see any adverse associations. In fact, we see protective associations for things like fruit, for yogurt, even sweetened dairy, for chocolate. Again, you see chocolate in all these cases. I told you I'd be your friend today. And for breakfast cereals. Interestingly, too, we see it for fruit juice, 100% fruit juice. So this is a fruit juice not behaving like a sugar-sweetened beverage, but behaving more like a fruit. We see a protective association you see there for stroke incidence, which we also see for hypertension incidence, which is, I think, interesting. If we look at the more patient-important, public health-important outcome, CV mortality, this is the big one, what do we see? We see a similar story. So we do see an adverse association for sugar-sweetened beverages, as I showed you there, but we don't see an adverse association for any of the other important food sources, and we actually see protective associations for a number of those. So again, for fruit, which we see quite consistently, for breakfast cereals, and for 100% fruit juice, we see a protective association. So here is a case where these important food sources of sugars are not showing an adverse association, in some cases actually showing protective associations. Now, what does the data from randomized, non-randomized trials tell us? So we've again started a series of systematic reviews and meta-analyses here. Some have been submitted. Nothing has been published, so this is all new data that I'm going to share with you. This has been sponsored principally through a grant that we received through Diabetes Canada. Here we actually have greater granularity in terms of the contribution of energy, because we have substitution trials where the sugars are swapped calorie for calorie in equicaloric comparison. We have the addition trials where the sugars or foods are added on top of the control diet without compared to the control diet without those excess calories. We also have subtraction trials. This is where the entry criteria for the trial is that they're consuming, let's say, a sugar-sweetened beverage, and then it's displaced those calories using water or low-calorie sweetened beverage. And then probably the most real-world and best example, which would be ad libitum trials, which is just free replacement under free living conditions. So I don't have time to go through all of the data. There's a lot of different food sources. We could do a whole seminar on this. So what I've done is I've summarized the signals that we've seen across these systematic reviews, meta-analyses. This represents 7 systematic reviews and meta-analyses, more than 500 trial comparisons, more than 10,000 individuals with follow-up from a week to a year. And what you can see here is we see the same thing, that it's really under addition conditions providing excess calories, and we may argue that's where the population is, so it's a very important place to look. We do see a contribution, but it really relates mostly to sugar-sweetened beverages, although we do see it for fruit juice, which suggests that although fruit juice is protective in the cohorts, we have to be careful about overconsumption. And we actually see some protective associations when those are removed, or protective effects, I should say, in the case there again of sugar-sweetened beverages. And we see some foods, such as fruit, for example, and I think we would hope to see that showing a protective effect under an isocaloric substitution. So that's the data. What I wanted to do very quickly, I have a few more minutes, I know I'm getting motioned here, is just go into the unintended consequences, and I think really the implications of these data as I see them, and I think they can inform the discussion. I think, if I summarize our data, what I would say is sugar-sweetened beverages appear to be the special case, and it's really under excess calorie conditions that they seem to manifest an adverse effect and have a negative influence through obesity. My concern is that if we drive the food industry to produce low-sugar foods, we may revisit the low-fat paradigm, where we replaced the fat with refined starches and sugars. The calories didn't change, and we didn't really improve public health outcome goals. And we could argue that things actually got worse. People may have regarded this as a license to eat as much as they like because these foods came off as having a health halo. My concern is that we do the same thing to sugars, and I think it's a very relevant question if that would happen, just because, again, the main substitution would be with refined starches. If you take the sugars necessarily out, given their functional properties in foods, necessarily the other components will rise to the top, if you like, and in most cases that backfill or that increase comes with refined starches. So you reduce sugars at the expense of increasing refined starches. And if I give you one example, and this is a cereal example, but we could also use other examples from cakes and crackers. And then this is one actually is drawn from my clinic where I had a discussion with a patient. I think that you can see this being relevant. So here we have two cereals. We have Kellogg Corn Flakes and Kellogg All Bran Buds with psyllium. If Kellogg is in the audience, my apologies. I didn't mean to pick on you, but this I think is a good example. I had a patient that came to me, and we're trying to counsel them on low cholesterol diets in lipid clinic, where we have portfolio of cholesterol lowering foods, in particular viscous fiber foods, mainly derived from hot and cold cereals. I wanted the patient to consume more All Bran Buds with psyllium. They were consuming cornflakes. I had another patient that was Special K, and they said, they were really concerned about sugar, so they were having cornflakes. But I said, I'd like you to try this one. He goes, well, it's got a lot of sugar. It's got 8 grams of sugar. I said, yeah, it's got 8 grams of sugar, but it has 13 grams of fiber, half of which is the viscous soluble fiber that will lower your cholesterol. And in fact, the calories are a little bit less because the fiber is displacing the refined starches, and it's got a lower glycemic index. So I think this is an example where if you were to choose your cereal based on sugars alone, you may be pushed away from even some of the higher whole grain or higher fiber options, and I think that that's an important consideration. The other one, I think, and Professor McGregor showed this in his presentation, I think it's very important, is it may distract us from all the other things that are important. We have to ask the question of all the things, all the risk factors, what ones do I need to worry about? And analysis that allows us to ask that question, a really important one, and Professor McGregor showed you an earlier, the 2013, this is the 2015 analysis, is from the Global Burden of Disease, which is a massive pooling project which allows one to compare across 79 risk factors and ask that question of all the things I need to worry about, what's important. Now, as Professor McGregor showed you, food and nutrition and food diet risk factors are the most important. But what about when we pull them apart? Are they all the same? And if we look here at sugars, at the only risk factor that is sugars containing, where high intake is associated with harm, which is sugar-sweetened beverages, and we ask ourselves, how does that compare to the other dietary risk factors? What you can see, and I'm just giving you the Canadian example, I pulled this out of 4,000 pages of the supplement. It's the least important of all the dietary risk factors we have to worry about. And if we look at other dietary risk factors that may be sugar-containing, and I've put those in gold, and a low intake is at risk. So it may be more important to increase those that are at low intake than decrease even the sugar-sweetened beverages at high intake. And if we look globally at the most important, the only risk factors that make the top 10 for dietary risk factors is whole grains, so I think it's important to promote those. And for the Western Europe, it's whole grain and fruit make the top 10. So my conclusion, and I share it with the authors, would be the same. This is the conclusion of the authors, is that if our findings are correct, a policy focus on the sugar and fat components of diets might have a comparatively smaller effect than that of promotion of increased uptake of vegetables, fruit, whole grains, nuts, and seeds. So again, I think we have to have some, nuance in how we approach this in terms of food. And I think one way to do that, and we're going to have that in the next session of discussion, is to shift more towards food and dietary pattern-based recommendations. This is something that's happening internationally and globally. It's something certainly that we're doing in Canada, and I give the Canadian example where we've done it with our Canadian Diabetes Association and Canadian Cardiovascular Society. What you can see here are a number of food patterns where you would have some sugar-containing foods that would be part of those healthy, evidence-based dietary patterns, but necessarily the sugars tend to be less in these, and for that matter, the sodium and other things, because when you eat the whole healthy dietary pattern to achieve the tenets of it, you necessarily reduce those things. So here are the takeaway messages. I think any effect of sugars appears to be highly dependent on the food source and comparator, so what are you going to replace it with, / energy control. Whereas we do have data for sugar-sweetened beverages showing an adverse association. The same does not hold for other important food sources, and there's even protective associations for some, such as yogurt, fruit, 100% fruit juice, whole grain cereals. I think dietary patterns will overcome a lot of this and allows us to take advantage of a lot of the interactions that occur within a complex plant matrix, so interaction between nutrients in the matrix and the foods. And I think then, so targeting sugars as a source of excess calories is very important, so I agree with Professor McGregor in that regard, but we cannot choose a healthy diet by sugars alone, and so little sugars can also help the low-fat dairy, whole grains, dietary fiber, and fruit to go down. So that's how I would like to leave it. Thank you.
Thank you very much to both speakers for the brilliant presentations and the very clear messages. We see a lot of overlapping as well, different points. But that's the purpose why we had this debate. So I will take the first question that we've got through the app, and then if someone would have a second question from the floor, because we'll have to move quickly to the next one. And then, as I said, we'll take all the questions, or almost all, at the end in the plenary discussion. So one of the questions submitted was, could you comment on the amount and quality of evidence grade which is available for other sugar sources than sugar-sweetened beverages? Can firm conclusions be drawn? Who's the first?
I guess I'll take it just because I showed those systematic reviews. We did do great. I didn't have a chance to go through it. Most of the evidence that we have is low to moderate quality in some cases. Some of the data for sugar-sweetened beverages is moderate quality because we upgrade it for a dose response and we don't downgrade it for any concerns. For a lot of the other food sources, for fruit we do have a dose response, for others we don't. So the evidence from certainly the cohorts tends to be low. In some cases moderate, and in terms of the trials there are a number of downgrades because there tend to be small trials with design issues. So I would have to say in general the evidence, and I think Dr. McGregor said this, is low. But I think it's equally low for all these things, so we still have to start somewhere.
Thank you. Would you agree, Professor McGregor?
Well, I mean, the problem, you see, coming from a background of blood pressure and controlled clinical trials with double-blind randomized trials, the problem about nutrition is that you can't get an answer and we end up with a mess. And I think my view is quite clear that we need a plan to prevent obesity. That's what we're talking about. We can go on about all the trials, this, that, and we need a plan to prevent type 2 diabetes. We need a plan to prevent high blood pressure. And we need a plan to reduce cholesterol. And I'm very pragmatic, and sugar is a major source of calories. I agree with the restrictions you have on reformulation. It's much more complicated to reformulate sugar than salt, and you need to reduce the calorie content, and we need to have fillers without calories, and I know someone from Nestle may talk about that later today. You can also use higher sugars, polyols, which in large doses cause diarrhea but are not metabolized. So it is possible to replace sugar and reduce calorie intake and reduce the sweetness. There's no doubt in my mind, although the evidence is appalling, that sweetness leads to increased appetite, just as saltiness does as well. And we need to think about that. But I think pragmatic solutions, it's no good arguing about all the difficulties about sugar, fat, and let's get on with a plan to prevent us getting obese, because people are dying now from obesity and type 2 diabetes, amputation, blindness, etc., renal failure, and it's a tragedy, and we can prevent it.
Okay, thank you very much. If there is a burning question from the audience, if not, okay, we have one. Please, the micro is coming. And very quick, please.
I'm the president of European Culinary Nutrition Academy, and our academy helps industry to lower sugars, fats, salt in the food. And we also higher fiber. I want to know, by your studies, how is important to higher the level of fiber in food, and with the increment of, with the lowering of fat, sugars, and salt, together with the...
Thank you for
I'll
the
start by
question.
saying I think that that's a real challenge, and that goes to my last point, which is if you produce a very high fiber food, let's say with 13 grams, if you try consuming that without sugar, good luck. It's like eating sawdust. So we need to be able to leverage sugar, fat, and salt where we can to get the healthy foods, but then we have to be careful not to do too much. But I think we want to promote those healthy foods. So in those foods, maybe we have to think about some exceptions, maybe, to the rule, if you like. For a sugar-sweetened beverage, I think it's a different story.
Yeah, I mean, I think, you've got, I agree, I don't disagree, you've got to be careful what you're doing. There's no point in taking out sugar and putting back, simple carbohydrates. You need to put complex vegetable residues back that have fiber in. And, they're obviously, if some companies are going to cheat, then we've got to deal with that and look at it. It is much more complicated than the salt reduction is very easy to do, and I don't know why every company is not doing it. Sugar is more complex, but it is possible to do it. I mean, and I can discuss that with you later, the technical problems of sugar reduction and getting a calorie reduction. Clearly, fat is much easier. As you all know, it has 2.5 times the calories of anything else. So you can take fat out and put back complex vegetable residues and have big reductions in the calorie content. And, many companies are now doing this, particularly Nestle and Unilever, are racing ahead because they see the writing on the wall. And in relation to your example of All Bran and Kellogg's cornflakes, they're both seriously unhealthy. All Bran has a huge amount of salt in and a huge amount of sugar. If you take shredded wheat, it has no added sugar, no added salt, and is full of fiber. So there are much better alternatives. And using Kellogg's cornflakes as an example, Kellogg's have the worst cereals in the world, and yet they put across that they're healthy, and it's a load of rubbish, and they can sue me if they want to.
Thank you very much for the answers. And I repeat, at the end, we'll take all the questions, and you'll have, again, the opportunity to interact with our guests. Thank you very much for the talks and for answering the questions. And we are moving now to the second debate about whether saturated fat causes chronic metabolic diseases. We have again 2 speakers, and they will present in the same way, and we'll have the same type of interaction. So the first speaker is Professor Philippe Legrand. He's professor and chairman of the Laboratory of Biochemistry and Human Nutrition in the Agronomic University of Rennes. And for more than 30 years, he performs research on fundamental aspects of fatty acid synthesis and metabolism. Since 1998, he's chairman of the French Guidelines Committee for the Fatty Acid Dietary Recommendations in the French Food Safety Agency. He's a member of the International Society for the Study of Fatty Acids and Lipids and member of the board of the French Nutrition Society. Thank you very much, Professor Philippe Legrand. And the second speaker on the same topic is Professor Ronald Mensink. He's professor of molecular nutrition in the Department of Human Biology, Faculty of Health, Medicine and Life Sciences at the Maastricht University. His primary research interests are in lipid metabolism, the relationship between nutritive and non-nutritive components in the diet with chronic metabolic stress, and particularly related to cardiovascular diseases. He carries out dietary intervention studies with the aim to provide evidence-based information in supporting the development of food, dietary guidelines and health claims. So please, come to the stage. Thank you very much for accepting the invitation, Professor Mensink. So you can sit wherever you want for the talk. And with this, I'm very happy to give the micro to Professor Philippe Legrand.
Thank you for the invitation. I would like the first slide, please. Maybe it's to me. So, saturated fatty acids have been for a long time presented as an homogeneous group of poisons. In fact, it's not an homogeneous group and it's not poisons. I'll try to demonstrate it and show that we need a new paradigm is necessary for consideration of nutrients before all. So we eat them, but we make them also, and in particular, one of them, which is palmitic acid. Indeed, from sugar, starch, alcohol, acetyl-CoA is directly entering the Wakil hélice to palmitic acid. And that's a general situation and everywhere in human animals and plants as well. Palmitic acid is the most fuel commonly found in all the nature. Without stop, except in the mammary gland, where short and middle chain saturated fats could be obtained just in this tissue. In terms of metabolism, they have not the same metabolism. There are plenty of difference inside the group of saturated fats. Beginning with absorption. As you probably know, long-chain fatty acids, saturated or unsaturated, are entering the organism through the formation of chylomicrons in the intestine and then lymph circulation and distribution everywhere. But short and middle chain fatty acids, and this occurs just for saturated, are entering directly through the portal vein and are beta-oxidized in the liver. So early here, so it's not the same fate in terms of absorption. When we, for instance, study the metabolism of saturated fatty acids, what we did since 10 or maybe 20 years, for instance, myristic acid, biosynthesis, gene regulation, acylation of proteins, desaturation, elongation, briefly, all the metabolism. We did that in comparison with palmitic acid, for instance. What are the lessons of this kind of study? Short and middle chain saturated fatty acids have a specific and safe metabolism. You cannot find them easily in the adipose tissue or in the arteries because they disappeared. Okay? Secondly, for instance, myristic acid, which is a C14, and palmitic acid, even with this small difference of 2 carbons, they have not at all the same fate in the cells. For instance, we extensively studied. Myristic acid is rapidly beta-oxidized, weakly secreted in the form of TG, but strongly elongated into palmitic acid, so no accumulation of myristic acid. On the contrary, palmitic acid is stored, secreted in the form of TG, weakly elongated into stearic acid, and is also, as I mentioned before, the main product of de novo lipogenesis from sugars, alcohol. It does accumulate in the cell. We could even call him sugaric acid, or fructosic acid, or alcoholic acid, maybe. So, very, very big difference in between. If we go on, stearic acid is less unsynthesized than palmitic, much, much less, actively desaturated into oleic acid. No accumulation also. So could we call it pre-oleic acid? So I want to blow up the group of saturated fat. They have important specific function. Here is a biochemist speaking. In addition to energetical function, here is a list from C4 to C24. C4, fuel for intestinal mucosa and microbiota, immunostimulation, cellular differentiation, and inhibition of tumor proliferation. If it was coming, and it is a case, from fiber fermentation, we would say, that's just marvelous. But he has no change. He is a group of horrible poison saturated fatty acids. So the group of C6, 8, and 10, obviously less fat deposition since they are oxidized in liver. C8, for instance, decreased VLDL secretion. It was written even in the case, first papers, inhibition of ApoB, hypocholesterolemia effect, and specific interesting functions like ghrelin acylation. Ghrelin is the only orexigene peptide, for instance. It's required for that, antifungal and antiviral properties. So that's interesting. Myristic acid, specific acylation of proteins. We studied that very much. What is acylation of proteins? It's fatty acid linking with a covalent bond to a protein, and to do this acylated protein, which is required to have the function of this. Around 100 proteins are known to be myristoylated. And as you can see, the functions obtained in this form of myristoylated protein, for instance, membrane association, possibility of anchoring in the membrane, subcellular localization, protein-protein interaction. For instance, here is a small unit of GDP protein anchoring in the membrane with two links, a myristic acid here and a palmitic acid here for obtaining the activation. The second point we demonstrate in our lab is activation of conversion for precursor in the omega-3 family. So you know probably we are lacking of DHA, but the pathway is very weak and not that active. In my lab, once we put myristic acid in the cell culture where we were studying delta 6 and delta 5 desaturase, and what we obtain is a significant increase of this pathway. So first in cellular, in the cell culture, then in rats, and then in humans. And finally, the presence of myristic acid for acylation of some proteins implied in the delta 6 complex led to increase EPA and DHA in certain form of lipids, phospholipids, for instance. So that's a nutritional point. We could say alpha-linolenic acid coming from oils, vegetable oils, plus minimum of dairy products bringing myristic acid leads to have more DHA, for instance. Activation of sphingolipids, just like structure, the myristylation is also required here at one step of the pathway for synthesis of sphingolipids, which are a part of membrane phospholipid. And you can see here in this pathway, palmitic acid is required as first substrate, second saturated fatty acid is required here, and myristic is required here to acylate the enzyme. And so, let's move to palmitic acid, component of sphingolipids, whatever, and phosphoglyceride. It also acylates some proteins for cell adhesion molecule, receptor and channels, and very important lung surfactant in the form of dipalmitoyl phosphatidylcholine, precursor of PEA, which is a neuroprotective and neuroanti-inflammatory effect. So plenty of good things. Stearic acid has also a function of sterilization of proteins, transferrin receptor, for instance, is actively desaturated to oleic acid, I say, and component also of phospholipids. These two guys are 1/3 of phospholipids fatty acid anyway, even in fish oils. And the third, here, nervous structure, myelinization. So what? Nutrients with important function first and not poisons, obviously. Let's move to problems with CVD and MS, metabolic syndrome biomarkers. So I am not an epidemiologist, but if we look at the RCTs with hard endpoints, even the most positive, Hooper, demonstrate no significativity on total mortality and all the rest. RCTs with surrogate endpoints by Mensing, effect of reduction, replacement on LDL, total 2-HDL, but not evidence of beneficial effects in terms of hard points. Translation from marker to CVD endpoints is an interesting debate. And replacement of part of saturated fatty acid by PUFA without any distinction of which saturated toward which polyunsaturated appears to me insufficient to demonstrate. So if we look at observational studies, I'll not comment, plenty of papers, but the recent meta-analysis like Siri-Tarino 2010 said no significant evidence for concluding that dietary saturated fat is associated and so on. The other one, which is often cited, is Chaudhary, same results, and other papers, but I don't want to comment that. Interesting is a general view obtained in the pure study, which could be criticized also. But you see here, total mortality in terms of saturated fatty acids shows that we are under the risk from 6 to 18% of energy. In major cardiovascular disease, it's also under the risk. And so, general problems, now that I have blew up the group of saturated fatty acids, we can say, no problem here with CVD risk. I can cite also the paper by Pragman and Mensing, obviously. Here, it was still correlated 10 years again, but now that it's not the case anymore if we consider total 2-HDL cholesterol, for instance. Palmitic, yes. Palmitic. Maybe it's not the bad guy. But it's not the bad guy by itself, because it's a nutrient with plenty of interesting functions. It's the bad guy by ourselves. So we eat probably too much, and we eat too much energy, and it's a marker of too much energy coming from carbohydrate, alcohol, and so on. And, and here, no problem. So, 1, maybe 2, but probably 1 is problematic. Let's conclude. No reason for considering saturated fatty acid en bloc anymore in terms of structure and metabolism. That's really old-fashioned. In terms of function, term of deleterious effect as well. Absence of evidence for specific deleterious effects. Need of more precise epidemiological studies. When considering intervention studies with substitution for oleic on N-6 polyunsaturated, not beneficial except substitution for N-3. So we should not say anymore polyunsaturated fatty acids for substitution. For instance, in my lab, this is in rat, okay, but you can see here in terms of inflammation, linoleic acid has the same inflammation power here than saturated fatty acid, and here saturated fatty acid on interleukin 1 beta is slightly more. But for instance here, with the transcription factor of inflammation NF-kappa, in aortic cells, in aorta, saturated fatty acid has a lower effects than linoleic acid. Just to say, even if it's physiology, that we have to be careful when speaking of replacement from saturate to polyunsaturated. We have maybe to say replacement of palmitic to omega-3, which are in deficit. And so intervention studies with substitution for carbohydrate, sugar, starch is not beneficial. Time for new paradigm and up-to-date recommendations without caricatural old statements of toxicity or eviction. And probably the new paradigm could, for instance, if we consider not only statistics and epidemiologists, but please put again some physiology in the panels, in the committees, and everywhere. Probably the new way to join molecules and nutrients between them is butyric acid plus fibers and probiotics. Well, here nothing. Probably here C14 myristic acid and precursor of omega-3. Here stearic acid plus oleic acid should be in the same group for the same maybe recommendation, instead of saying it's a wall between saturated and the rest. And what is left is palmitic acid. Problem, yes, in case of excess, I mentioned already, C6 palmitic or sugaric acid from fructose could be joined with fructose, glucose, alcohol. Maybe that's the reason we are all four invited today. With total energy, obviously, and maybe with linoleic acid also. Need of a balanced and synergic recommendation for palmitic acid would be a big, big progress in the field of saturated. So we try in France through the recommendations of the Food Safety Agency and CES, but it's quite old already. We did that in 2010, saying what? We blow up the group of saturated. The blowing was modest because here we were still joining lauric plus myristic plus palmitic acid because they were still correlated with LDL to HDL. And as I mentioned, the two first disappeared in between, but these are the recommendations from 2010. But we applied the cautious and quite severe 8% for this group, saying if some are dangerous, so let's apply the severity to this. That was a big progress already, and not to the total group. So 8% here, and we consider that for the total here, there was place to 2% of short and middle chain for an omnivorous. That means short and middle chain are coming mainly from dairy products, for instance, or cocoa oil. And this is 2. And 2% place for stearic acid, which was at this time, and it's true now also, replacement possible for trans fatty acid, for instance, and 2%. And that's made 10%. And if we will do it again now, that's my personal opinion, we will keep 8% but apply to palmitic only, and 12% for the rest. Thanks for your attention.
much, Professor Legrand. And it's now time to listen to Professor Mensik. Please.
Okay, so this is already my, I think my second slide, but we need to speed up anyway, so that's okay. Some research funding, committees I'm part of, and the topic of today. So I've been asked to talk about chronic metabolic diseases as related to saturated fat intake, and if you focus on chronic metabolic diseases, and it has been said before, you acknowledge that there are many metabolic diseases. I mentioned the metabolic syndrome, cardiovascular disease, dyslipidemia, etc., etc. So we cannot discuss them all, and I made a selection also partly based on my expertise on cardiovascular disease, dyslipidemia, and a little bit of NAFLD, non-alcoholic fatty liver disease. If we talk about saturated fatty acids, and it has been said before, they are present in all foods. So there's not one single food that does not contain saturated fatty acids. The intakes in general are poorly reflected by body lipid pools. So if you eat a lot of myristic or palmitic acid, it is poorly reflected in body lipid pools. An exception may be C15, C17, saturated fatty acids from dairy that are not synthesized by the human body, and they are reflected by the body lipid pools to some extent. As has been discussed before, we do synthesize them ourselves, and there are many different saturated fatty acids, and MCTs, lauric, myristic, palmitic, stearic acid, and here you see some important sources of these fatty acids. But what is the actual intake of these different saturated fatty acids? Here are some studies that looked into the intakes, and actually there are not that many studies looking at the intake of the individual saturated fatty acids. C12, 14, 16, 18, and total saturated fatty acids. And what you see in 3 different cohorts is that the intake of lauric acid is quite low, 1-2%. Myristic acid 2-3%, and the major intake is of palmitic acid, about 5-8%. And stearic acid is normally about 1/3 of palmitic acid intake, so it is a little bit below 3%, 4%. If we look at dietary guidelines to lower coronary heart disease, and I would like to mention that I'm talking about coronary heart disease. There are many types of cardiovascular disease, but they are not all related to saturated fat intake. Coronary heart disease is the type of cardiovascular disease that is related to saturated fat intake. And that is mainly focused on lowering LDL cholesterol, irrespective of the country, the United States, Europe, the Netherlands, France, etc., etc. The focus is on lowering LDL cholesterol. Why? Because we think LDL is causally related to coronary heart disease. And there is many studies, genetic studies, animal studies, intervention studies, drug studies, etc., showing the higher the LDL, the higher the risk for coronary heart disease. So we accept that LDL cholesterol is a causal risk factor for coronary heart disease. If we look at dietary recommendations, there are not that many studies with disease endpoints. So we rely on biomarkers, and one of these biomarkers is LDL. So the idea is, does saturated fat intake increase LDL? Yes. Is LDL a risk factor for coronary heart disease? Yes. And then include it into dietary guidelines. And I think that all these questions can be answered with yes, and I will show you some evidence for it. One of the studies, and that is the meta-analysis we have done, that we looked at the effect of saturated mono, poly, and also trans fatty acids, but I will not discuss them now, on the effects on LDL cholesterol. And what is important here is that the effects are expressed relative to carbohydrates. So carbohydrates is set at 0 because you exchange. You cannot add saturated fatty acids to the diet, it has been mentioned before, because energy intake will increase, obesity will increase, your LDL will increase, not related to saturated fat intake, but due to overconsumption of energy. So carbohydrates were the point of reference. So what was found? That saturated fatty acid as a group increased LDL cholesterol compared to carbohydrates. Cis, MUFA, so oleic acid, did decrease it. PUFA, linoleic acid, did decrease it. And the effects of cis-PUFA were a little bit more favorable than those of oleic acid. But saturated fatty acid, the mixture of saturated fatty acids, there's a fly here, but the mixture of saturated fatty acids definitely increase LDL compared to carbohydrates. But you can make the same graph not using carbohydrates as point of reference, but using polyunsaturated fatty acids as point of reference. Here you see the effect of saturates if you exchange them for polyunsaturates. This is oleic acid, this is carbohydrates, and then you see that cis-PUFA has the strongest decreasing effect compared to the other macronutrients on LDL. And cis-PUFA in these studies are mainly linoleic acid. And this is more or less the sum of this plus that. So the effects are additive. So cis-SAFA plus cis-PUFA is the same magnitude because PUFA here is the point of reference. So when you interpret the results, be aware that the point of reference is. As said, the effects of the saturated fatty acids may differ. Here you see C12, 14, 16 and C18, again compared to carbohydrates. And what you do see is that C14 has the strongest effect, but at the same time you need to remember that the intake of myristic acid is the lowest. C16 has a huge effect. And stearic acid has hardly any effect, again, compared to carbohydrates. But compared to polyunsaturated fatty acids, it will increase LDL cholesterol. So the point of reference is really important when you discuss the effects of saturated fatty acids or of any macronutrient on LDL cholesterol. Also, when we look at intervention studies replacing saturated fats by another macronutrient, then you do see a decrease in coronary heart disease. And this is the meta-analysis of Hooper. Saturated fats were replaced by polyunsaturated fats. You do see a decrease. No studies on replacement by monounsaturated fatty acids, and some studies replacing saturated by carb or protein, hardly any effect. So from this meta-analysis based on randomized controlled trials, you can conclude replacement saturated fat, again by cis-PUFA, may have a beneficial effect, again on coronary heart disease. But still there's some confusion about the relation between saturated fat intake and cardiovascular disease, coronary heart disease. So statements like saturated fat do not increase heart disease, we need to revisit all the guidelines, etc., etc. But is that really true? I think that may not be true. So what causes the confusion? The confusion is caused by that several prospective epidemiological studies and meta-analysis based on these prospective studies have not shown an association or relation between the intake of saturated fat with coronary heart disease. But I think we can expect that such an association is not evident. Also, other studies in the field of nutrition did have some contradictory findings if you look at prospective cohort studies versus randomized controlled trials. Antioxidants are a clear example. We thought they were really protective, but the first intervention study was terminated because of adverse effects of antioxidants. Folic acid is a bit of a point of discussion, and I think the same for saturated fatty acids. So what are the reasons for these poor associations between saturated fat intake and coronary heart disease? I think one of the reasons is that it is difficult to estimate food intake, nutrient intake. I listed some sources of variation. I will not mention them all, but it is really, really difficult to get an idea of what are people eating. And for saturated fat, it has been estimated if you want to know the intake at the individual level of saturated fat intake, you need 22 randomly collected 24-hour dietary recalls. And many epidemiological studies only used 1 or 2 days, or food frequency questionnaires, etc. So I think that it is really difficult to estimate the intake of saturated fat at the individual level. Another issue is that we need to realize that diet is not the major determinant of LDL. There are other determinants of LDL, and if we follow the link saturated fat intake, LDL, coronary heart disease, and diet is not the most important determinant of LDL, you can see that the association is weakened. And I try to explain, and I hope I can make it clear, a little bit in this slide. Here you have 9 subjects or 9 groups, whatever you like. And this is really hypothetical. This is the non-diet part of someone's LDL. And this is the LDL caused by saturated fat intake. For this subject, it is the same, and for this one is the same. But what you can see is that the non-diet is the same for these subjects, but the total LDL is still lower for these 3 than for these and for these. So the risk for coronary heart disease may increase for these subjects based on their LDL, but if you group them based on saturated fat intake, subject 1, 4, and 7 will be one group and have really, really different LDL cholesterol levels. And that makes it difficult to look cross-sectionally at the relation and association between saturated fat intake and coronary heart disease. I think for these types of questions that randomized trials are more powerful. Also, we can dispute, okay, what are the actual lipid risk markers, biomarkers to predict coronary heart disease? LDL is one, but also you have effects of the macronutrients on triacyl glycerol, LP-little-a, and name it, but also on other risk markers as well. So what do we need to do? How can we make a step forward? And it has also been discussed. We need to think in substitution scenarios. And this is, I think, a study published 3 years ago, and they substituted saturated fats, for example, by trans, mono, PUFA carbs, and they did differentiate. And what they did see, okay, well, if you substitute it by trans, it increases. If you, by MUFA, you see a decrease, PUFA a decrease. So you replace one macronutrient by the other. So no isolated statement about saturated fat, because that is not helpful. It is always compared to something else. So again, substitution is, I think, the magic word. Another final issue that I would like to address, it is not simply saturated fat and LDL. One of the other studies for example, is that saturated fat may also affect other risk markers of disease. And in one recent publication, they looked at the overconsumption of saturated fat and looked what happened with the amount of fat in the liver. If it increases, you get many, many different diseases, obesity, type 2, atherogenic dyslipidemia, etc., etc., etc. So if the amount of fat in the liver increases, that is not considered that beneficial. So what did they do in that study? They overfed, and that is important, so people were not in energy balance. People with saturated fat, and what is interesting here, that the source of saturated fat was coconut oil and butter. So especially C12, 14 are important saturated fatty acids from these types of oils. Saturated, unsaturated, and carbohydrates. And they looked, okay, what happens with the amount of liver fat? And what you do see is when people are not in energy balance, and I think many, many people are not in energy balance, that saturated fats increase intrahepatic triglycerides more than unsaturated and carbohydrates do. So that shows that saturated fats not only affect LDL by one mechanism or another, but also have other unbeneficial metabolic effects. So I think that are enough arguments to say, okay, we do need an upper level for the intake of saturated fat, and we cannot leave it free and not limit the intake. So saturated fats are present in all foods and needed by the body. I think I do really agree with the previous speaker, but I think there is convincing evidence that diets low in saturated fat and at the same time high in cis, unsaturated fatty acids, substitution again, lowers coronary heart disease. No doubt that different saturated fatty acids have different metabolic effects, but the question is it already time to translate these findings into dietary recommendations? If you look at these studies on hepatic fat, you did see that coconut oil and butter, which are not that rich in palmitic and stearic acid, did increase liver fat. So I'm not really sure it is already time to make the distinction, but I certainly see the arguments. Do not only focus on coronary heart disease, there is more. We need to discuss substitution scenarios, and what will be discussed, I think, after the break, nutrients, foods, and food patterns. That is also an important issue to address. Thank you for your attention.
Thank you very much. Thank you very much, Professor Mensik. Now we will take only one question from the audience. If we have, through the app, we didn't get anything specific to saturated fats for now. But please, as I said, send your questions because we will have time in the panel discussion at the end to address. Is there any question from the audience, or if the two speakers would like to ask the other one something very specific? No? Yeah, I see that everyone wants to have a coffee and a break. So we will come back at 10 o'clock. Thank you very much to everyone.